All right, let's jump right in. Ready for a deep dive.
Let's do it.
Today, we're diving into the world of, you guessed it, microscopic worms.
Here we go again.
Well, more specifically, C. elegans, and how scientists are using some seriously cool tech, nanosensors, to study their digestive systems.
And it's pretty remarkable stuff. C. elegans is already a star in the research world.
Yeah, their short lifespan, transparent body. I mean, talk about a living laboratory.
Exactly. And this research takes things to a whole new level. But before we get too deep into it, maybe a quick recap of what makes C. elegans such a valuable model organism.
Yeah, good idea. I think for anyone new to the world of C. elegans, a little background would be helpful.
So imagine a living, breathing laboratory, but just a millimeter long. That's C. elegans. Short lifespan, transparent body. You can actually see what's happening inside, especially with these new nanosensors.
Oh, yeah, those nanosensors. That's what we're really here for. But just to finish up with C. elegans, you were going to say something about their genetics.
Oh, right. Their genetics are incredibly well studied. Really helps us understand how genes affect biology. It's kind of amazing.
Okay, so we've got these tiny worms with their whole lives on display. And then enter the nanosensors. What makes these things so special?
These nanosensors are like, well, imagine tiny pH meters.
Tiny pH meters, so they measure acidity.
Yeah, but operating inside a living organism, they're designed to light up differently depending on the acidity of their surroundings.
Okay, I think I'm starting to get the picture. But how do they work? I mean, I'm picturing something like a tiny glowing probe. Is that close?
You're not far off. They're made of this porous material packed with special molecules called fluorophores.
Fluorophores.
You can think of them like tiny fireflies that emit light. Some of these fluorophores are sensitive to pH, so their brightness changes based on the acidity level.
Oh, that is clever. But how do you even get the nanosensors inside the worms in the first place?
Well, it took some clever maneuvering. The researchers basically mixed the nanosensors with C. elegans food, usually bacteria, E. coli, typically.
Ah, right, E. coli. So the worms ate the bacteria with the sensors mixed in, and boom, nanosensors are in.
Yeah, pretty much.
Okay, nanosensors are in. Now what? How do researchers track them? How do they get those pH readings? They're not taking microscopic blood samples, are they?
Not quite. Remember how we talked about the fluorophores, how they emit light?
Right, tiny fireflies.
Exactly. So scientists can use special microscopes that detect this light.
Ah, I see. So they create images of where the nanosensors are and how their brightness changes.
Yeah, and that allows them to create a detailed pH map of the worm's digestive system. Pretty cool, right?
So they're essentially watching the worm digest in real time. That's amazing. Did they find anything surprising?
Oh, absolutely. They discovered a distinct pH gradient along the digestive tract. The most acidic area in the posterior intestine had a pH of 3.59.
Wow, that's seriously acidic. I mean, why so harsh?
Well, think of it this way. The acidic environment helps break down food efficiently. You know, those digestive enzymes we talked about? They work best in an acidic environment, like a chemical cauldron for digestion.
Makes sense. So it's all about making sure the worms get the most out of their bacteria buffet.
Exactly. And it gets even more interesting. The most acidic region, it also happens to be where most of the proton pumps are located.
Proton pumps.
They transport protons, which directly influences the pH level. So there's this clear link. Proton pumps, acidity, and efficient digestion.
It makes you wonder if this system is similar in other organisms.
Absolutely. That's one of the exciting things about this research. By studying C. elegans, we might learn more about how digestion works in other organisms, even humans.
Wow, I never thought I'd compare my digestive system to a microscopic worm's. But C. elegans eats bacteria. Our systems are way more complex, right?
Sure, our system's more complex in some ways, but at a fundamental level, the basic principles are remarkably similar.
So even though we're separated by millions of years of evolution, we still share some basic digestive strategies with these tiny worms. That's incredible.
It really is. And there's so much more to uncover.
Well, we'll definitely dive deeper into all of that after a quick break.
And here's where the real-time aspect gets really interesting. We're not just talking about a static snapshot of pH levels. These sensors let researchers track those changes as they happen, you know, like capturing the dynamic fluctuations in acidity as the worm digests.
Oh, that's right. You mentioned the worm's pharynx pumping incredibly fast. Didn't the researchers have to, come up with some clever tricks to capture those rapid changes?
You've got a good memory. The C. elegans pharynx pumps at a mind-blowing 300 contractions per minute. Imagine trying to get a clear picture of something moving that fast.
It'd be a blur, so how they slow things down?
They took advantage of the fact that C. elegans is cold-blooded. By lowering the temperature, they could slow down the worm's metabolism, and that slowed down the pumping rate of the pharynx. At 4 degrees C, the pumping rate dropped to a much more manageable 7 contractions per minute.
So they basically hit the pause button on digestion. That's brilliant. What do they see when they slow things down?
Rhythmic pH oscillations, fluctuations in the intestine, particularly in the anterior region.
Oh, right where the pharynx connects to the intestine.
Exactly. That's where the pharyngeal contents are being delivered.
It's like the first stop on the digestive train line, and things are already getting acidic. Did they see these fluctuations anywhere else?
Further down the intestine, yeah. They saw similar pH oscillations, but they were delayed compared to the ones happening right at the pharyngeal intestinal junction. Suggests a carefully orchestrated wave of acidity.
So it's not just one big acidic bath. It's like a carefully timed sequence of pH changes.
Right.
What's the reason for that?
It's likely due to the peristaltic movements of the intestine, those wave-like muscle contractions that help push food through. This wave of acidity likely helps optimize the breakdown of food as it moves along.
It's amazing how much complexity is packed into such a tiny creature. Now, thinking bigger picture, how does this research on C. elegans digestion relate to our understanding of human digestion? Are there any real parallels?
More than you might think. While our digestive system is certainly more complex than a worm's, the fundamental principles are surprisingly similar.
So even though we're separated by millions of years of evolution, we still share some basic digestive strategies with these tiny worms.
Exactly. And this is where things get really exciting. For instance, remember those proton pumps we talked about? They play a key role in regulating pH in both the C. elegans gut and our own stomachs.
Yeah, and you mentioned problems with those pumps can lead to conditions like acid reflux. Could this research help us develop better treatments for those kinds of digestive disorders?
That's the hope. By studying how these pumps function in a simpler organism like C. elegans, researchers might uncover new targets for therapies that could benefit humans.
It's incredible to think that studying these tiny worms could have such big implications for human health. Is there anything else this research might shed light on?
Well, another fascinating area is the role of gut bacteria. We know that our microbiome, the trillions of bacteria living in our gut, is crucial for digestion and overall health. And guess what? C. elegans also relies on bacteria as its main food source.
So could studying how C. elegans interacts with its gut bacteria provide insights into our own microbiome?
That's a really interesting possibility. Imagine being able to manipulate the pH levels in the C. elegans gut using those nanosensors and then seeing how those changes affect the microbiome. It could be a powerful tool for understanding how our own gut bacteria are influenced by factors like diet and medication.
It sounds like this research is opening up a whole new world of possibilities for understanding digestion and its impact on health. But I have to ask, are there any downsides to manipulating these biological systems? What are the potential risks?
That's a crucial question and one that researchers are very aware of. While the potential benefits of this technology are enormous, it's essential to proceed cautiously and ethically.
Right, it's important to weigh the potential benefits against any unforeseen consequences.
Exactly. But for now, let's focus on the incredible insights that this research has already provided. We've seen how these tiny transparent worms are helping us unravel the mysteries of digestion. We've explored the role of pH, the importance of proton pumps, and the potential impact on human health. But there's still one piece of the puzzle we haven't touched on: the groundbreaking implications of manipulating pH levels in the C. elegans gut. We'll dive into that fascinating territory after.
Oh, actually, you know what? We've got time. Let's just go ahead and jump right into that now.
Yeah. Okay, so let's talk about manipulating those pH levels. Things get really interesting here. Remember those nanosensors? And how they rely on fluorophores that emit light.
Yeah, the tiny fireflies.
Right. Well, some fluorophores can do more than just measure pH. They can actually change it.
Wait, hold on. So we're not just watching the digestive process anymore, we're actually taking control.
Exactly. It's called optogenetics, and it's revolutionizing biology.
Optogenetics.
By shining specific wavelength of light on these special fluorophores, researchers can trigger chemical reactions that alter the pH in very precise locations.
So they can basically dial up or down the acidity in different parts of the worm's gut. That's incredible. But why? What can they learn from that?
Well, think about it. They could investigate how different pH levels affect those digestive enzymes, or how changes in pH impact the gut microbiome.
It's like having a microscopic control panel for digestion. What are some of the potential applications for C. elegans research and for human health?
Well, for C. elegans, researchers could study specific digestive conditions like acid reflux in a really controlled way.
Ah, I see. And that could lead to a better understanding of those diseases.
Right. And potentially new treatment strategies.
And what about for human health? Could this technology eventually be used to treat those conditions in people?
It's definitely a possibility. Imagine like light-activated therapies to adjust the pH in our own digestive systems.
It sounds like science fiction. But it also raises some ethical questions, right? I mean, if we can control something as fundamental as pH, where do we draw the line?
You're absolutely right. As with any powerful technology, there are ethical considerations. We need to make sure it's used responsibly for the benefit of all.
It's a good reminder that scientific progress comes with responsibilities. But for now, I'm still blown away by the possibilities this research has opened up.
It really shows the power of curiosity, the amazing things we can discover when we delve into the seemingly ordinary. Who would have thought that studying a microscopic worm's digestive system could lead to such groundbreaking insights?
It's a perfect example of how sometimes the smallest organisms reveal the biggest secrets. So we've seen how scientists are using these cutting-edge nanosensors to map and even manipulate those acidity levels in the C. elegans digestive system. Not only is this research giving us a deeper understanding of digestion in general, but also opening up new avenues for research into human health.
And this is just the beginning. There's still so much more to explore. Who knows what other secrets these tiny worms hold?
Well, for anyone fascinated by the microscopic world and the complexity of even the simplest creatures, keep those microscopes handy. And if you want to delve deeper into this research, we've got a link to the original paper in our show notes. And remember, sometimes the smallest organisms reveal the biggest secrets. Until next time, keep diving deep.